All right, now today our lab is going to cover enzymes. We are doing lab number 5, starting on page 33 of the lab manual. We're really going to start on page 34 because page 33 is mostly just questions. Our major research question today is essentially what factors influence the rate of an enzymatic reaction. And in fact, there's a more specific research question for the day on the top of page 38 that you can look at once we get there. Does the concentration of substrate affect catalase activity is a more precise research question for today. So we'll have to define some of those things as we go. So here's kind of an outline. We're going to have some background, of course. We're going to work on our experimental setup and write a hypothesis. You're going to design your own experiment more or less today. And then your assignments for today are going to be to continue to work on your lab report on osmosis. And so that's according to, if you look at the manual, it says no more than 2 pages. So I'm expecting about a 1.5 page paper or so there. And then a lab summary is what the actual assignment for today is. A lab summary you've not done before. I posted it on the lab website for everyone who's watching this video, but for my students it's also on Blackboard. It's basically, it says what's your hypothesis, what's your figure, put your figure here, and then what's your conclusion based on this. So that's what a summary is. It's like a mini lab report, okay? And so that's what the assignment will be for today. So we have not, at least for my sections, we've not quite gotten to this in lecture yet, so let's give a little background. So this lab is about enzymes. And what are enzymes? Well, enzymes are proteins typically. So we have primary structure made of amino acids folded up into a secondary structure, folded up into a tertiary structure, held together with lots of bonds. And then we might even have quaternary structure, where we have several chains of amino acids folded up to tertiary structure and stuck together. These enzymes have very specific shapes, and one area of an enzyme is known as the active site. The active site is the part that binds to another molecule that we're just going to call the substrate. Substrates bind to the active site, and they're held in there with hydrogen and ionic bonds, things like that. And when they stick to the enzyme kind of hugs it, kind of changes shape a little bit. And you might have read about induced fit in your book. That's induced fit. It means it's not quite like a lock and key, but the enzyme actually changes shape when the substrate docks with it. And they call this the enzyme substrate complex, which is behind my video here because of the video. But that's just the enzyme stuck to the substrate. Now, what that's going to do, if you've read any of the chapter yet, that's going to lower the activation energy needed to get a reaction to take place, which means you can get a chemical reaction to take place at body temperature or room temperature instead of having to heat something up with a Bunsen burner to get it to happen, which is important. You don't want to have to run a fever of boiling in order to get your chemical reactions to take place in your body. So what this does is it lowers activation energy mostly in 2 ways that we'll worry about. And these are 2 important ways that you might want to take note of because students always forget to put it in their second lab report that you may well be doing over enzymes in a couple weeks. They do it in 2 ways. One, they stress bonds, which allows the substrate to break apart at lower temperatures than if you didn't have the enzyme around. So they stress bonds, allowing them to break. Or, so that's like if you're breaking something apart like we're doing today. Or they bring substrates together with the proper orientation for a reaction. Those are the two major ways that we'll worry about today, how enzymes work. Now let me try to give you an analogy to what we're talking about here. So the first one, let's say this marker is the substrate and your body really wants it to come apart, the lid, the cap to come off, right? You're trying to break it. One way to do that in like a lab is to put it in really hot water, and that breaks bonds and makes things come apart. That's not really an option for an organism. So one way that an enzyme might work is this substrate comes to the active site, pressure is put, and this cap is able to come off with a lot less energy than would be needed without the enzyme. Okay? The other way this could work, let's say we have two substrates and you're trying to build something. An enzyme might dock to both of them and put them together and create whatever the next molecule is in this reaction. Okay, so we've created a product here. You can do this in chemistry too without an enzyme. If you have molecules floating in water, remember they move, right? We talked about diffusion, osmosis, and all. They're moving around. They will bump into each other and react. What would you have to do to make things bump into each other really fast and react in a test tube, in a chemistry lab? How do you increase the rate at which things bump into each other? Increase the temperature. That means the molecules are moving more and more and more, and they're slamming into each other more and more and more. And you get this to happen more often. But with an enzyme, you don't have to increase the temperature necessarily. You can just put a lot of enzyme in there, and it will bind to each one and bring them together. Now, of course, temperature will still have an effect on the reaction, because the faster or the warmer it is, at least to a point, the more often these will then bump into the active site and then be able to be put together. So temperature will have an effect on these reactions as well, and that'll be what the next lab is about. Assuming, I'll put a big caveat on there, assuming I can find enough hydrogen peroxide at the store to keep running our labs like we have planned, because that's one of the items that has been hoarded a lot lately is peroxide. So if I can find enough peroxide, we'll do this lab for 2 straight weeks. Well, you'll have a week off, but you know what I mean. All right, so here is what we often refer to as the catalytic cycle of enzymes. There's an important lesson to be seen in this diagram. So let's start at number 1. Substrates enter the active site. In this case, we're looking at like number 2 here, where substrates are brought together in the proper orientation by the enzyme. They dock to the active site, forming the enzyme substrate complex. Now they dock, and the enzyme probably changes shape, and they're brought together at just the right angle for the reaction to take place. So that's what this says. Activation energy is lowered, basically, so the reaction speeds up because there's not as much energy needed to make it happen. Then substrates are converted to products. The products are released. Now here's the important thing to see here. The active site is now available for more substrates. So the enzyme is not part of the reaction. It's not a reactant. It doesn't get used up. It's more like when you're building a house and you have a hammer. You build the house, and when you're done, you still have the hammer, right? It can be used over and over again. So that's something I want to point out. That reminds me, we're going to do a demo in a minute that I need to get some things out of the fridge for. Would one of the TAs run to the fridge and grab a lovely container of liver for me, and a cutting board? There's a drawer marked cutting boards. Thank you. So that's how enzymes work in general. Now let's talk about the specific enzyme for today. Now this enzyme has a name that sometimes confuses students. So let's start with definitions here. First of all, enzymes are catalysts, right? Catalysts lower the activation energy needed for a reaction. Enzymes are organic catalysts made by organisms. Now the specific enzyme we're using today is called catalase, which sounds a little bit like catalyst, and that's where sometimes folks get confused. Okay? The enzyme we're talking about is called catalase. It is found in your cells Well, several places, but it's concentrated in your peroxisomes. One of those organelles sort of at the end of the cell chapter that we covered a week or two ago. Peroxisomes. So what it is it's a tetramer of 4 polypeptide chains. What do you think that means? You playing at home can answer too. What do you think that means? Tetra means what? 4. So there's 4 polypeptide chains here. So do you think this has only tertiary structure, or does it have quaternary structure? So if you remember, what is primary structure of a protein? By definition, what is it? The order of what? We can't flush exam 1 content out of our minds just because we move past it. Primary structure. The order of what? What are proteins made out of? Amino acids. So the order of amino acids is primary structure. There's 20 different amino acids. Which ones are where? That's primary. You fold that up into an alpha helix or a beta pleated sheet. That's secondary structure. You fold that up again and form a globular protein, which is like there, globular protein, okay? I mean, there'll be a specific shape there, but it's a glob, okay? Globular protein. That's tertiary structure. What do I need to do to make quaternary structure? Another one of these. So I need another totally separate primary structure that's folded up into a blob, and then these two stick together. That's quaternary structure, okay? Some proteins only have tertiary structure, but this one has 4 separate polypeptide chains. So 4 tertiary structures stuck together to make catalase. So what it does is it protects your cells from oxidative damage. What I mean by that is that during some of our cellular respiration and other processes, sometimes we make, especially I think when we break down fats, we tend to make hydrogen peroxide. And hydrogen peroxide is not something you really want to have in high quantities in your cells. Hydrogen peroxide tends to oxidize things. It tends to nab electrons. It does damage. I mean, hydrogen peroxide can be used as rocket fuel or a component of rocket fuel. It's not something you want in quantity in your cells. So we have an enzyme called catalase. So we make peroxide, but we also have an enzyme that helps to break it apart real fast too. So one catalase enzyme can convert millions, and I've seen numbers like 40 million. I have no idea how it's that fast, but it's really fast. Hydrogen peroxide molecules. Hydrogen peroxide is H2O2. It can break it down to H2O, which is a really bad joke. Like 2 guys go into a bar, and the first guy says, "I'll have H2O," and the second guy says, "I'll have H2O too," and then he died. Anyway, so strong peroxide is actually quite bad for you. The stuff we get at the store is only 3%. It's not too dangerous. So one catalase enzyme can convert millions per second. This is the active site. Kind of looks like a Pac-Man mouth or something. I had a picture behind my behind the video here of you can actually buy Catalase at Walmart sometimes. They sell it as a pill that's supposed to prevent your hair from going gray, because they claim that when you get older you make more peroxide or something in your hair follicles, and that's what bleaches your hair gray. But we'll talk maybe later about why that's probably not something you could fix with a pill. But here's the reaction. 2 hydrogen peroxides in the presence of catalase will yield 2 water molecules and an O2, oxygen, molecular oxygen. Everyone who's ever had a grandma pour peroxide on their cut knows this. What does peroxide do when you put it on a cut? It bubbles. That's the production of the oxygen gas. When you pour it down a drain, has anyone ever poured peroxide down a drain? You hear it go, pfft, you hear the hissing? That's because it's popping the membranes. We've done that in another lab with a different substance, but it's popping membranes of bacteria in the drain, and it's reacting with their catalase. And so it's being converted to water and oxygen gas. So every living thing that I know of, anyway, that lives in an environment with oxygen in it has catalase. It's a very conserved enzyme. It's found in almost all life forms that I know of that are exposed to oxygen, because peroxide is occasionally generated and needs to be broken down. So the question today is, does the concentration of substrate affect catalase activity? And to be even more specific, as I said earlier, there's a more specific, and maybe I should just change this slide, but I haven't yet. A more specific question on the top of page 38. You know, does the concentration? Well, actually, no. Actually, I lied. The wording is the same on the top of page 38. Now, what is the substrate today? The substrate of catalase, the thing that docks to catalase and reacts. H2O2, or hydrogen peroxide. Thank you. Yes. So ultimately, you could say, does the concentration of hydrogen peroxide affect catalase activity, if you wanted to remind yourself what the substrate is. But that's what we're going for today. All right. So I'm going to do that demo in just a second. I'm going to have to get a knife or a pair of scissors out. What we're going to do is we're going to do a demo with some liver to see this reaction in action and talk about it. And then we're going to get ready and set up our experiment. But I'm going to go ahead and do most of this video here first, and then we'll do the demo, okay? Maybe we can do one of the demos on the video. That'd be exciting. I'm sorry that the blinds are up, but I'll just read to you what it says here. But this is the basic experimental setup today. We have a probe on top of a stand. That is a probe that records pressure. Atmospheric pressure. It has a stopper at one end that will fit into test tubes. Okay? You also have a stir plate at your bench, which is that black box. And there should be a stir bar on top of it, a little magnetic bar. What's going to happen is you're going to make up your own yeast solutions in a little bit. You're going to get yeast from the back of the room, make your own stock solution to bring back to your bench. You're going to drop a stir bar into that yeast solution and set that container on the stir plate. And as that stir plate runs, it'll spin the magnet inside your flask, and it'll keep the yeast stirred up. Because yeast can't swim, so they have to be stirred to keep them in suspension. So that's what that is. If you've never used one before, ask a TA or me for a little guidance as you set that up. Okay, so a few suggestions for today before I move on is be sure to clean all your glassware, like rinse it out before you use it, just in case someone didn't clean it out very well, because sometimes students leave the stuff contaminated, and then we get the chemical reactions happening before we even try to collect data, and you get nothing and it goes nowhere, and it's frustrating. And we're going to need to wear goggles today, and we should wear gloves as well. Not that 3% peroxide is real dangerous, but just to keep you safe. And then sometimes test tubes explode in this lab, not terribly commonly, but they have, and we don't want to get glass in our eyes. So the basic idea is when we put yeast and peroxide together in this test tube, we're using yeast as the organism today, the model organism. Because yeast is a eukaryote, just like you. It has all the same organelles you have, including peroxisomes. And those peroxisomes contain catalase. So yeast is the organism. It's a fungus. It's just bread yeast that you get at the store that we're using. It's a fungus. It's a single-celled organism that contains peroxisomes just like you, which have catalase inside, this enzyme that we're studying. So we're going to use them because it's readily available. It's easy to maintain in the lab. Although it's another thing being hoarded right now because everyone's suddenly baking bread during the pandemic. So another thing that this lab's double whammied in terms of like stuff that's always super easy to find at the store and now is really hard to find at the store, oddly enough. So what I'm going to have you do while I get the liver ready for the demo is I want someone to log in to the computer By the way, how about you two work together? And who wants a third person at their bench to help out? It's a lab. Okay, you don't mind. Just maintain as much. You might stay where you are for now to maintain distance, but an extra set of hands won't hurt today anyway. So, all right. So what I want you to do is to log into the computer. Let me just show you how this hooks up. This LabPro unit here, I tried to hook them all up earlier. Hopefully they're all on and working. We'll find out. But the probe here plugs into channel 1 on the side of this thing. Like this one doesn't have any lights on right now, which concerns me slightly, but we'll see what happens. Oh, there it goes. Stay on. Anyway, what I want you to do is to make sure your probes are plugged into the blue box here in channel 1. And I want you to go to the lab website, and under today's lab, you'll see this link that says Logger Pro Template. I want you to download that and open it, and that will open up Logger Pro with all the right settings for the lab today, assuming it works the way I hope. Okay? So while you're doing that, I'm going to set up, get the liver demo ready, and I'm going to pause this recording so people don't have to watch a half hour of nothing. For those of you watching at home, we're about to squirt peroxide into a baggie with chicken liver in it. We're going to watch the reaction. Go, you ready? There you go. Now seal the bag up. Now see if you feel it. Like feel the bag where the reaction's going, and see if you notice anything qualitative. We're looking for qualitative observation. Okay, what do we notice from a qualitative standpoint? What's it look like when you put peroxide in there? What's it do? Bubbles. Bubbles. Did you notice anything else? Any other qualitative observations? Did anyone feel it?
Feels like it's
It might break some of the tissues down. It actually releases heat and it will warm up. It's an exothermic reaction. It'll release heat as it breaks things down. And that'll be something we talk about. We'll talk about like exergonic reactions in lecture. It's a reaction that breaks things down and releases heat. Now here's a question. What do you think would happen now if you added more peroxide? If you added another 5 mils, what do you expect would happen? This is something I normally have you do, but since I'm rationing peroxide, we'll probably just talk about it. But what would happen if you added another burst of peroxide? Would it react again or not? What do y'all think?
It would just keep breaking it down a little bit more.
Okay. Any thoughts back here? What would it do if you added another 5 mil of peroxide? Okay, so some students, a common misconception that I'll often hear when I ask this question is some students will say it won't do anything because the catalase is used up. Do you use up an enzyme in a reaction? No, the enzyme's still there, so it would bubble again. It might bubble a little less because you have a bunch of water in there now, so things will be diluted, but it would bubble again. So this is the... You can do this at Thanksgiving this year if you go home and there's some like turkey innards laying around. You can be like, hey, everybody, watch this, and squirt it with peroxide and talk about enzymatic reactions. Why don't y'all do me a favor now and throw away those baggies before they accidentally leak all over our notebooks and stuff. So that's the basic experiment. Now, once you have that file downloaded and opened, it should open Logger Pro, and it should be reading in units that are millimeters of mercury, which is like mmHg. The way we used to measure atmospheric pressure was by how many millimeters the pressure could push a column of mercury. So that became a unit. And even though we're using no mercury, we're using electronics, we still use that unit. Does everyone see that unit on their screen? Is it working? Yeah? Okay. Excellent. All right, now I'm going to give you some basic tips today to survive and do well. Keep your peroxide and yeast pipette separate and labeled. So if you have a red one, just make that your peroxide pipette. Then have another one, maybe keep it on the other side of the lab bench that's for the yeast, so that way you don't mix them up. Because if you mix them up, then the reaction takes place in the pipette and you get no data from the probe, and that's bad. I'm going to ask that we just did it once, but we're going to not pipette out of the stock container. What I'll have you do is figure out how much stock you need, and you can pipette it out of the stock container when you get everything you need for the day, but put it in a beaker, and then pipette out of that for the rest of the day. That way we don't overuse the peroxide, and if we do mess up and we accidentally get yeast in the peroxide, it'll be in a cup of peroxide and not the whole container. Here's a big one. Do not wash your stir bar down the drain. Students often clean up and they forget there's a little magnetic stir bar in their cup, and then it whoop, goes down the drain. Those are like stupid expensive for some reason, so don't wash it down the drain. What you can do is catch it in that little strainer that you used last time to catch your potato bits. Just catch it when you're done. And I put a thread up here of -10 points if you put your stir bar down the drain. So I want to see your stir bar stuck back to your stir plate at the end of the day. Wear protective gear at all times. Also, when you put your stopper on your test tubes, don't hold it on the test tube. The stopper can pop off when pressure builds. It'll go pop like a cork gun and it'll pop off, but that's good because if you're holding the test tube, the stopper on, then the bottom of the test tube explodes instead. We don't want to do that. And don't hold or shake the test tubes because that adds heat from your hand, which changes the variability of the results. We're adding more variables. And it adds motion, which increases the rate of the reaction also. So all the reactions are going to take place in those test tube stands that you have. I see one there, you got one there. Hopefully they're all out. The reactions will take place in those stands. Okay. Now, your manual talks about on the top of page, well, near the top of page 37, it says to prepare a stock yeast solution, which is your source of catalase. So let's talk about how you're going to prepare that, just so you know. And I'll turn you loose to do it in just a little bit. You're going to add 40 mils of distilled water to a clean plastic beaker and 3 grams of dry yeast. 3 grams of dry yeast. And so we have balances around the room. We have 3 of them out. And we have some weigh paper and weigh boats out, so you can measure out 3 grams, add it to that water. Then you're going to put the stir bar in there and then turn it on your stir plate. Now when it's on your stir plate, you don't need to crank it to all the way fast. It'll look like a tornado then. And you want it to have a nice dimple in the water and everything to be stirred, but you don't need to go all the way to 11. Okay? So that's how you'll make that. Then what we're going to do several things here. I'm going to walk you through the first run of the experiment so you can see how the equipment works. Then I'm going to have you write your own hypothesis after we see how everything works. And then what you're going to do is you're going to make your own dilution. You're going to come up with what dilutions you're going to make. You're going to make your dilutions today. So you're going to test 5 different concentrations of peroxide today. One of them is going to be 0, control, right? And one of them is going to be full 3% straight out of the bottle. And each one of those concentrations you're going to test 3 times. So you're going to do your own replications, okay? And you're going to look at the rate of the reaction. So we're looking at rates today, rates of reactions. Instead of just saying what's the pressure, what's the millimeters of mercury, we're going to be looking at millimeters of mercury per second. That's a rate, like miles per hour. Not just how far did you go, but how fast did you get there. So we're asking how fast is the reaction going is really what we're measuring today, okay? So we'll talk all about that as we go. I just don't want to make this video too long here. So when we get our data today, I'll explain this in a moment, but the data is going to look kind of like this when you on the screen of your computer. We're going to get the slope of this line that you see here, but we're only going to get the initial slope before the part of this line that's linear. So like, and for this one, it's like right here, and then you can see it starts to slow down and curve off. We're only going to get the slope of that first linear part, the initial rate of the reaction, because it's going to start slowing down because it's making a bunch of water and it's basically diluting itself as it runs, and it's going to slow itself down for a number of reasons as products accumulate, so on and so forth. So we only want that initial reaction. So we're going to look at how altering substrate concentration affects the rate of the reaction. And so your basic graph is going to look like this. You're going to have hydrogen peroxide concentration. That's your independent variable. And your dependent variable will be catalase activity, which will be millimeters of mercury per second. So what you're measuring is how much oxygen gas pressure is being created as that reaction takes place. Okay, so we did the liver demo. What we're going to do now is you're going to make your yeast stock solution. Then we're going to do a demo of how the equipment works without using any yeast or peroxide, just so I can show you that when you put the cork on, it changes the pressure of the test tube just from the cork going on, and we don't want to record that in our data, so I want to show you what that looks like. Then we're going to do one all together using 3% peroxide, just straight peroxide. We're going to do an initial run of that experiment so you can see how the equipment works. Then you'll work on your hypothesis and design and run your experiment. We're going to do a minimum, let's just say, I don't expect you to go over that today, but you're going to do 5 concentrations of peroxide and 3 replications. So each one you're going to do 3 times. And I'm going to show you what that data table needs to look like so you know how to keep track of your numbers. So we're not collecting class data today. You're going to use your table's data, okay? So then you'll design and run that experiment, then you'll clean up, and then you'll be ready to go, okay? So that's the plan, the ultimate plan. So we'll get to I'll just pause this. This is complicated filming and pausing, but let me, this is take 2. Okay, so hypothesis writing. We're going to use a research hypothesis which has 3 parts, if, and then. The if part is the actual hypothesis. If you look up hypothesis, a hypothesis is by definition a tentative explanation, meaning your best shot at explaining what you think is happening, how these 2 variables interact, what's going on. So if, and that's your hypothesis, and then you put and that's what you're going to do. And then is followed by your prediction, what you think the data will look like if your explanation is correct and you do these things. That's what a research hypothesis is. It has those 3 parts. For example, if hydrogen ions disrupt hydrogen bonds and catalase is exposed to a pH gradient, then as the pH decreases from neutral, catalase activity will decrease. So you could say, so your if is going to be what you think the relationship is between activity and catalase, or activity and hydrogen peroxide concentration in this case. So the example I gave you here is like if instead of testing substrate concentration, we expose catalase to different pHs to see if pH messed up the enzyme, right? And so you have the if, and you have what you think pH does to enzymes, and what you're going to do, and then what you expect you'll see. So in this case, let's talk about this hypothesis. What will adding substrate do, having a higher concentration of substrate? What is that going to do for the reaction? Let me put it this way. Let's say my hand here, this Pac-Man thing, this is the active site. Molecules are always in motion, right? We can speed up a reaction maybe by warming it up. We talked about that. That would increase the activity of substrate meeting the active site. But how do you think concentration of substrate will affect the rate of the reaction? What will having more substrate in that test tube do? Will that cause more of them to react at a time, or more of them to hit the active site more frequently if there's more of it? So there's probably a relationship there between how much substrate is floating around and how often they're going to bump into the active site. So when you do that if part, if you want to talk about it like you're in the magic school bus or you have a super powerful microscope and you can see the molecules. If increasing substrate concentration causes substrate to more frequently impact the active site. That would be an explanation, right? And tell them what you're going to do, and then what you think the data will look like if you're correct. Now there is one thing, because we haven't done a lot of this, I'm going to point out you may or may not see this in your data. I'm kind of out of room here. I'm going to erase the data collection table. You might see either a real or a statistical plateau in your data, which you'll have to explain if it happens. So you might, but you might not, see your data do this, or if it stays linear, this marker's dying, if it stays linear, sometimes you'll have data points here with a little error bar, but this error bar up here is huge, let's say. This is what I would call a statistical plateau, meaning the averages did go up, but the variability is huge here and it overlaps with this one. So if you see that, you might have to explain why that happened based on how enzymes operate. But I'll look at what your data does, and I want to hear from you what your data did before you leave today so we can get an idea of what happened, okay? So you might see a plateau, and if that happens, you can predict that in a hypothesis if you want, but if it does happen, you'll have to interpret why it happened. That's the conclusion part of the assignment, is why did your data happen? Explain it, okay? All right, so I'm going to let you make your concentrations and let you just run your reactions. You know how it works now. You know how the equipment works. You're going to run it. You're going to get the slope of the first 4 or 5 seconds of that data, or if there's a really obvious linear part, you get the slope of that part. And then you write that down, and that's all there is to it. Then you clean up when you're done, okay? Have at it. And thanks for watching at home. Don't forget to play the at-home game. I don't know if there's anything else nope, that's it for today.